Far below the surface of the ocean lies the farthest edge of our planet, a place where depth becomes distance and distance becomes pressure. Beneath the waves, the ocean stretches wider than any continent, and yet most of it remains unseen, unmapped, and unvisited. Sunlight fades within the first few hundred meters. Beyond that gentle boundary, the world begins to change. Familiar reference points disappear, replaced by water, stone, and time. Pressure rises steadily with depth, enough to overwhelm most machines and reshape materials we think of as solid. The seafloor becomes less a place you stand on and more a place you endure. Here, sound travels farther than light ever could. Vibrations bend and move across entire basins, carrying information through darkness. Instruments record signals that echo for hours, sometimes for days, without a clear source. Expeditions descend with careful plans and often return with fragments, partial images, brief recordings that resist simple explanation. Despite these conditions, life continues, not powered by sunlight, but by chemistry. Entire ecosystems survive on heat, minerals, and reactions that would poison most surface life. Some species are observed only once, captured briefly on camera, and never seen again, leaving questions instead of answers. Tonight, we are diving into the deepest parts of our ocean to examine the deep sea mysteries that are still keeping scientists awake at night. But before we begin, if these deep sea journeys help you relax, I would appreciate it if you leave a gentle like or even subscribe to this channel. And if you feel like sharing, let me know in the comments where you're listening from and what time it is there. It's a joy to see how far these sleepy explorations travel. Now, take a slow breath, close your eyes if you wish, and let's begin. Long before a vessel ever leaves the harbor, the challenge has already begun. Blueprints are studied under quiet laboratory lights, materials are tested beyond their limits, and engineers attempt to predict forces that cannot be fully rehearsed at the surface. The descent itself is only the visible portion of an effort shaped by constraint. As depth increases, pressure builds with unrelenting steadiness. It presses inward on hulls, joints, lenses, and seals, searching for the smallest imperfection. Metals flex microscopically, transparent viewports cloud or fracture. Electronics sealed inside protective chambers must endure forces measured in thousands of pounds per square inch. Many designs function only once before deformation renders them unreliable. Each journey downward carries the understanding that equipment may not return unchanged. Light offers no assistance. Below a certain threshold, illumination must be carried from above, and artificial beams reveal only narrow corridors of water at a time. Beyond those cones of brightness lies an expanse that absorbs vision completely. Pilots rely on sonar reflections and instrument readouts, constructing a sense of space from echoes and shifting lines on a screen. Movement becomes deliberate, cautious, guided more by interpretation than by sight. Cold compounds the difficulty. Temperatures hover near freezing across vast stretches of seafloor. Batteries lose efficiency. Hydraulic fluids thicken. Mechanical arms respond more slowly than they would in warmer seas. Even the most advanced remotely operated vehicles feel the drag of this environment, their energy reserves diminishing with every minute spent below. Communication threads stretch thin across miles of water. Fiber optic cables relay images when conditions allow, yet signals can flicker or degrade. Acoustic transmissions travel far but arrive delayed, refracted by layers of varying density. Instructions sent from a support ship may take seconds to reach a vehicle, and those seconds expand in significance when maneuvering close to fragile terrain. Real time becomes an approximation rather than a certainty. Time itself is scarce. A descent may consume hours before a vehicle reaches its target depth, leaving only a brief window for observation or sampling. Weather at the surface can shorten missions unexpectedly. Mechanical concerns can force an early ascent. What is gathered often amounts to fragments, short recordings, limited samples, impressions shaped by narrow opportunity. Returning to the exact same coordinates is rarely simple. Deep currents shift sediment, submarine landslides alter contours, hydrothermal activity builds and collapses mineral towers. Even without dramatic events, the ocean floor evolves quietly, reshaping the context in which earlier data was collected. A site mapped one year may appear subtly different the next, complicating comparison. All of this unfolds within financial and logistical boundaries. Research vessels operate on tight schedules. Each expedition represents years of planning and substantial cost. As a result, exploration advances in increments, patient and methodical. Knowledge accumulates slowly, assembled from scattered dives and partial glimpses, a mosaic built piece by careful piece beneath the weight of the sea. There are moments when the deep ocean speaks without revealing itself, not through movement or light, but through vibration. Far from shore, hydrophones rest in the water column, suspended in quiet suspension, listening to distances that human ears could never endure. They do not search for spectacle, they wait. Sound behaves differently in the sea. Layers of temperature and salinity create shifting corridors where acoustic energy bends and travels, sometimes trapped within channels that carry it across entire basins. A pulse released in one hemisphere can be detected thousands of miles away, arriving altered yet intact. In this world, vibration becomes the most faithful traveler. Many of the signals recorded are familiar to trained analysts. The grinding fracture of ice, the low tremor of submarine earthquakes, the rhythmic churn of distant vessels. Patterns emerge, are cataloged, and folded into growing archives. Yet threaded among them are other sounds, singular and resistant to easy placement. Some arrive as elongated moans that stretch beyond a minute, rising and falling in pitch with a smooth, organic quality. Others are sharp and percussive, echoing through the water in rapid succession. A few repeat over days, maintaining consistent intervals before fading from record as abruptly as they appeared. Certain frequencies exceed the expected range of known marine mammals. Others fall within biological limits, yet lack the modulation typical of communication or navigation. The ocean itself participates in shaping these signals. Seamounts reflect and scatter vibrations. Deep trenches focus energy in unexpected directions. Thermal layers refract acoustic paths, altering both intensity and direction. What begins as a localized event can emerge on a recording device transformed by distance and geology, its origin obscured by the very medium that carried it. Researchers approach such anomalies with patience. Waveforms are compared against seismic databases. Spectral analyses examine harmonic structure. Models simulate how known animals might sound under varying pressure and depth. Geological activity is cross-referenced with timestamps. Often a plausible explanation surfaces, subtle and grounded in physics. At other times, classification remains tentative. There are recordings preserved in institutional archives that still carry provisional labels, not because they suggest something extraordinary, but because available data cannot yet anchor them to a clear source. The duration was unusual, the amplitude unexpected, the repetition pattern incomplete. Without a visual encounter or corroborating measurement, certainty remains out of reach. In this way, the deep ocean communicates in fragments. Hydrophones continue their quiet vigil, capturing the steady language of shifting crust and migrating life, alongside tones that resist translation. Each signal becomes part of a broader acoustic tapestry, a reminder that even in darkness, the planet is never silent, only speaking in dialects still being learned. On certain stretches of the ocean floor, the water shimmers as if it were rising from an unseen fire. Columns of mineral-rich fluid surge upward through fractures in the crust, carrying heat from the planet's interior into surrounding darkness. Around these vents, life gathers. There is no sunlight here to anchor a familiar food web. Instead, microscopic organisms draw energy from chemical reactions. Hydrogen sulfide, methane, dissolved metals, substances lethal to many surface forms, become nourishment. Through chemosynthesis, bacteria convert these compounds into organic matter, forming the foundation of entire communities. Tubeworms cluster in pale forests. Mussels and clams anchor themselves in dense beds. Shrimp drift through rising currents, their bodies adapted to a world that would overwhelm most known physiology. Temperature gradients can be extreme within the span of a few centimeters. Vent fluids may exceed 300 degrees Celsius before mixing rapidly with near freezing seawater. Organisms occupy narrow margins where conditions are neither too hot nor too cold, neither too acidic nor too depleted. Proteins within their cells remain stable under pressure that would distort ordinary molecular structures. Membranes maintain integrity despite chemical concentrations that disrupt typical metabolic pathways. Life persists by adjusting its chemistry to match its surroundings rather than resisting them. Some vent fields remain active for decades. Others fall silent when underlying magma shifts or mineral conduits clog. When activity ceases, communities collapse or disperse, dependent as they are on a constant chemical supply. New vents form elsewhere along mid-ocean ridges and back arc basins, and larvae carried by deep currents must locate these rare habitats to survive. The distances between them can span hundreds of kilometers, making each ecosystem feel both connected and astonishingly isolated. Beyond hydrothermal systems, cold seeps release hydrocarbons from buried sediments. Here, too, microbial life forms the base of intricate networks. Carbon compounds that once lay locked beneath layers of rock rise slowly and become sustenance. Crustaceans, anemones, and specialized fish settle nearby, their existence tied to invisible flows from below. How such ecosystems first emerged remains a subject of careful study. Some researchers consider whether similar chemical environments on early Earth provided the stage for life's origin. Mineral surfaces within vents may have offered catalytic structures, concentrating molecules and fostering complex reactions long before photosynthesis reshaped the planet. Others emphasize gradual adaptation, envisioning surface organisms migrating downward over evolutionary timescales, adjusting generation by generation to deep conditions. In many of these habitats, genetic lineages differ markedly from shallow relatives. Barriers of distance and depth limit exchange. Each site evolves with subtle independence, shaped by local chemistry and geology. The result is a mosaic of biological innovation, sustained without reliance on the sun, rooted instead in the restless energy of Earth itself. A camera lowers through the water with steady patience, its lens open to whatever may drift across its narrow field of view. Hours pass in quiet observation, sediment rising and settling in slow motion. Then, without announcement, something unfamiliar enters the frame. It may appear for only seconds, a translucent body moving with delicate pulses, a long filament trailing behind, like a thought half-formed. The shape does not align neatly with known families. Its proportions seem slightly out of balance with expectation. The vehicle shifts to follow, but the creature drifts beyond the edge of illumination and is gone. Back at the surface, the recording is replayed again and again. Analysts pause on single frames, enlarging details, tracing outlines, comparing features to archived species. Sometimes the footage is grainy, disturbed by particulate matter or limited resolution. A mechanical tremor may blur the image at a crucial moment. No physical specimen rests in a container for careful measurement. Only light captured on a sensor remains. Many deep-sea organisms are fragile beyond assumption. Gelatinous bodies collapse when brought upward, their structures supported not by rigid skeletons but by the surrounding pressure of their habitat. Even with pressurized recovery chambers, tissues can degrade rapidly. Enzymes begin breaking down cells once conditions shift. By the time a sample reaches laboratory benches, its defining features may already be altered. In some cases, a single sighting stands alone for decades. No second encounter confirms its range or abundance. Without repeated observation, population size remains unknowable. Behavior cannot be mapped across seasons. Diet and reproduction are inferred from morphology, from subtle hints in body design. Classification may remain provisional, a tentative placement within a broader lineage. There are organisms known only from damaged nets hauled up from great depth, their bodies torn or incomplete. Others exist solely as faint silhouettes in remotely operated vehicle footage, never close enough for detailed inspection. Genetic material cannot be sequenced without tissue. Evolutionary relationships remain speculative, sketched lightly in scientific literature with cautious language. It is possible that some of these forms inhabit narrow ecological niches, their distribution limited to small pockets of seafloor or midwater columns rarely surveyed. It is also possible that certain species pass through life cycles that make them visible only briefly, emerging at depths where cameras seldom linger. The ocean's vastness allows both rarity and concealment. Occasionally, a creature first seen once is rediscovered years later, its existence confirmed and expanded upon. More often, the record remains solitary, a few seconds of movement, a shape against dark water. Within archives, these moments rest quietly, reminders that documentation does not always equal understanding, and that some lives unfold beyond repeated witness. In certain corners of the deep ocean, scale shifts in subtle but unmistakable ways. A crab appears with legs spanning wider than expected. An isopod resembles its shoreline relatives, yet expands to the size of a small plate. A squid's eye seems almost disproportionate, round and immense, designed to gather the faintest trace of movement. The proportions of life stretch quietly beyond familiar boundaries. This pattern, often called deep-sea gigantism, has been observed across multiple groups. Amphipods grow larger than their shallow counterparts. Sea spiders extend delicate limbs across surprising distances. Some species of jellyfish and cephalopods reach dimensions rarely seen closer to the surface. The enlargement is not universal, yet it recurs often enough to invite careful study. Cold plays a role. In low temperatures, metabolic rates slow. Energy is used sparingly. Growth unfolds over longer spans of time, and lifespans can extend far beyond those of related species in warmer water. Cells divide at measured pace. Development stretches across years, sometimes decades. In such conditions, bodies may continue increasing in size long after surface relatives have reached maturity. Pressure, too, shapes physiology. Proteins and cellular membranes adapt structurally to maintain function under immense force. These adaptations can influence tissue density and fluid balance, subtly altering how organisms build and sustain mass. With fewer rapid predators and less competition in some habitats, evolutionary pressures differ from those in sunlit zones. Size may accumulate gradually, generation after generation. Yet gigantism does not appear everywhere. Many deep-sea creatures remain small, even minute. Some lineages display no clear trend toward enlargement. The pattern is selective, varying by region, depth, and evolutionary history. Availability of food plays a complex part. In certain areas, organic material drifts downward in sparse pulses, favoring organisms that can store energy efficiently. In others, localized resources allow sustained growth. No single explanation accounts for every case. Large eyes offer one practical advantage in near total darkness, gathering scarce photons produced by distant organisms. Extended limbs may increase the likelihood of encountering food in an environment where contact is rare. Greater body mass can store reserves through long intervals between meals. At the same time, larger size demands more energy, creating a balance that must be maintained through careful physiological tuning. Genetic studies continue to compare deep species with their shallow relatives, tracing divergences across millions of years. Some expansions in size appear to have occurred independently in separate lineages, suggesting similar environmental influences acting repeatedly. Others seem tied to ancient adaptations unique to specific groups. In the quiet expanses below, scale becomes another variable in the experiment of survival. Some bodies stretch toward unusual dimensions, while others remain modest. The darkness does not dictate a single outcome. It provides conditions within which life adjusts, sometimes expanding beyond expectation, sometimes remaining restrained, always shaped by the measured pace of the deep. In the midwater column, far from any shore and beyond the reach of dawn, brief constellations appear and dissolve. A pulse glows, then fades. A ribbon of blue arcs through the dark, followed by a scatter of smaller sparks. Here, illumination is not borrowed from above, it is made. Bioluminescence shapes much of life at depth. Through internal chemical reactions, organisms produce photons with remarkable efficiency. In many species, the process relies on luciferin, a light-emitting molecule, reacting with an enzyme called luciferase. Oxygen participates, energy is released, and a glow unfolds without generating significant heat. The effect can be steady, rhythmic, or instantaneous, depending on the design of the body that carries it. Some animals release light into the surrounding water as a form of defense. A sudden flash can startle a predator or create a luminous cloud that distracts attention long enough for escape. Certain shrimp expel glowing secretions, forming a temporary veil. Others detach small bioluminescent particles that continue shining as they drift away, drawing focus elsewhere. Predators employ light with equal precision. Anglerfish extend delicate lures, tipped with glowing bacteria housed in specialized organs. The faint beacon draws curious prey within reach. In the open water, hatchetfish produce controlled light along their bellies, adjusting intensity to match faint residual glow from above, rendering their silhouettes nearly invisible when viewed from below. This strategy, known as counter-illumination, erases contrast in a realm where shadows are rare but detection remains constant. Communication also unfolds through light. Squid and certain fish display patterns across their bodies, activating clusters of photophores in coordinated sequences. Flashes may ripple from head to tail, forming signals whose meanings remain partially understood. Courtship, territorial display, and schooling behavior appear to involve luminous exchanges too subtle for human eyes to decode fully. Not all species share the same biochemical pathway. Some generate light through intrinsic reactions within their own cells. Others rely on symbiotic bacteria housed in dedicated chambers, cultivating microorganisms that emit a steady glow in exchange for nutrients and protection. The diversity of mechanisms suggests multiple evolutionary origins, arising independently in separate lineages over vast spans of time. Why bioluminescence has become so widespread in the deep sea is still a matter of study. The darkness provides a canvas where even the smallest emission carries significance. Light can attract, repel, conceal, or reveal. In an environment where vision operates on narrow margins, the ability to create illumination becomes a versatile tool. Across these depths, living light flickers continuously, not in grand displays but in measured signals woven into daily survival. The sea at night, which is nearly all night here, is threaded with these quiet radiances, each one a brief declaration of presence within a world that otherwise remains unseen. Occasionally, during a routine survey, a shape crosses the view of a submersible that does not match the expectations of the mission. It is cataloged, measured against reference scales, noted carefully in logs. The encounter feels ordinary in its procedure, even if the organism itself is unfamiliar. Then, in the years that follow, nothing. Research vessels return to the same coordinates. Transects are repeated along similar depths. Cameras drift through comparable layers of water, yet the species recorded before does not reappear. Databases hold a single cluster of observations, sometimes from one expedition alone, separated by decades from any similar record. These absences do not always suggest rarity in the simple sense. Some deep-sea organisms may inhabit narrow vertical bands within the water column, rising and sinking in patterns tied to subtle environmental cues. Temperature shifts, measured in fractions of a degree, changes in dissolved oxygen, or variations in particulate food descending from above can alter distribution. If surveys occur outside these windows, presence remains undetected. In other cases, life cycles unfold at a pace unfamiliar to surface biology. Certain invertebrates grow slowly, mature late, and reproduce infrequently. A generation may span many years. If reproductive pulses are synchronized across large areas, population density could fluctuate dramatically over extended intervals. A survey might coincide with a peak in visibility, followed by long periods when individuals remain dispersed or confined to habitats beyond routine observation. Geographic isolation also complicates understanding. Deep basins separated by ridges or currents may function as semi-independent systems. A species abundant in one region could remain absent from another for centuries until gradual dispersal bridges the divide. Without continuous monitoring across vast distances, such movements pass largely unseen. There are recorded instances of fish and invertebrates documented in the early 20th century, then absent from literature until rediscovered generations later. Advances in imaging technology have occasionally brought back into view organisms once presumed lost to record. Yet, for every rediscovery, there remain entries that stand alone, their dates fixed, their locations precise, their follow-up unresolved. Environmental triggers remain difficult to identify. Chemical signals, subtle shifts in pressure gradients, or long-term climatic oscillations may influence emergence. Deep ocean circulation operates on timescales extending beyond human careers, redistributing water masses and the life within them gradually. Population cycles could therefore unfold over centuries, rising and falling in rhythms that exceed the span of systematic study. Within the archives of marine institutes, charts and specimen logs hold these gaps quietly. They do not signal disappearance in every case, nor certainty of persistence. They mark intervals where observation and existence fail to overlap. In a realm measured in patience, appearance and absence can both be temporary, unfolding according to patterns still taking shape beneath the surface. Along certain stretches of the seafloor, symmetry emerges in places where randomness might be expected. Blocks of stone appear aligned in repeating angles. Columns rise with edges that seem almost measured. Ridges intersect at near right angles, forming outlines that suggest intention rather than accident. The impression can be striking, even through the distortion of a remote camera. These formations often lie in regions shaped by volcanic activity or shifting tectonic plates. When molten rock cools slowly beneath the surface, it contracts in organized patterns. Basalt can fracture into polygonal columns, each face fitting against the next in tight geometric arrangement. What appears deliberate may instead be the quiet mathematics of thermal stress unfolding over millennia. Sedimentary layers add another dimension to this illusion. Over time, deposits of mineral-rich particles settle in horizontal bands. Pressure compacts them into stone, preserving distinct strata that can later be exposed by underwater landslides or currents. When erosion removes softer layers unevenly, sharper edges remain. Terraces form, sometimes stacked in sequences that resemble steps or platforms. Tectonic uplift and faulting further complicate the landscape. Plates grind slowly against one another, lifting sections of crust while lowering others. Fracture lines propagate outward, creating linear boundaries that cut cleanly across older rock. In areas where multiple stresses converge, the resulting geometry can appear almost architectural, a network of straight segments and angular corners shaped not by tools but by force distributed through solid earth. Carbonate structures contribute their own patterns. In some places, mineral-rich fluids seep through sediments, precipitating limestone or other deposits that harden into clustered towers or walls. Over time, these accumulations can take on repeating forms, guided by chemical gradients and subtle variations in flow. The shapes may appear regular from certain perspectives, yet closer inspection reveals irregularities consistent with natural growth. Despite plausible geological explanations, limited access leaves many details unresolved. High-resolution mapping requires equipment that can withstand depth and remain stable across uneven terrain. Lighting conditions affect perception, casting shadows that exaggerate straight lines or flatten curves. A structure photographed from one angle may appear different when approached from another, yet revisiting exact coordinates can be difficult in dynamic underwater settings. As a result, interpretations remain careful. Researchers compare rock samples, analyze mineral composition, and model stress patterns in laboratory simulations. Most formations align with known processes of cooling, compression, and erosion. Still, each new discovery is approached with attention, acknowledging that seafloor geology continues to reveal variations not fully cataloged. In the deep ocean, natural forces operate over spans of time that encourage order as often as chaos. Geometry does not always require intention. It can arise from pressure, temperature, and chemistry acting with steady persistence. What first appears designed may simply reflect the quiet precision embedded within Earth's own mechanisms. Far beneath the rolling surface, mountain ranges rise that no horizon has ever held. Their peaks do not pierce the sky. Instead, they press upward into miles of water, forming chains of submarine volcanoes that outnumber those on land. Many remain unnamed, mapped only as subtle elevations in bathymetric data, their contours traced by passing instruments. These volcanoes form where tectonic plates diverge or converge, allowing magma to ascend through fractures in the crust. Along mid-ocean ridges, molten rock wells up steadily, creating new seafloor as it cools. In subduction zones, descending plates melt and feed volcanic arcs hidden beneath the waves. The processes are continuous, yet direct observation is rare. Eruptions often unfold without witness. Seismic sensors may register tremors, and satellites sometimes detect slight changes in sea surface height or temperature, but the events themselves occur in darkness under immense pressure. Lava behaves differently here. Instead of explosive fountains driven by expanding gases, magma extruded at depth cools rapidly, forming rounded pillow basalts that stack gently upon one another. The surrounding water absorbs heat with efficient silence. Even without dramatic explosions, these eruptions release vast quantities of energy and dissolved minerals. Iron, manganese, sulfur, and other elements enter the surrounding water, altering local chemistry. Plumes of heated fluid can rise hundreds of meters before dispersing, carrying trace metals across considerable distances. Over time, such contributions influence nutrient distribution and microbial activity in ways still being quantified. Where heat escapes persistently, ecosystems gather, echoing earlier glimpses of life sustained by chemical gradients. Microorganisms colonize fresh volcanic rock within days or weeks, forming biofilms that begin the slow transformation of sterile surfaces into living substrate. As mineral-rich fluids circulate, larger organisms may settle nearby, their presence tied to the steady warmth and altered chemistry provided by volcanic activity. Deep eruptions also reshape the terrain. Lava flows bury existing habitats, while new fissures open pathways for hydrothermal circulation. Seamounts can collapse partially, sending debris downslope in slow avalanches. Each event modifies the physical structure of the seafloor, creating a mosaic of ages and textures layered upon one another. Understanding these processes requires a combination of remote sensing, sampling, and long-term monitoring stations that can withstand harsh conditions. Yet the scale of submarine volcanism far exceeds the capacity for continuous observation. Thousands of sites remain unvisited. Many eruptions pass unrecorded, inferred only from fresh rock or subtle chemical signatures detected later. Beneath the ocean, volcanic systems operate with steady persistence. They build new crust, redistribute elements, and provide energy to organisms adapted to their presence. Much of their influence unfolds quietly, integrated into the broader dynamics of the planet. In this concealed landscape, fire and water coexist, shaping a world that remains largely beyond direct view. On certain parts of the ocean floor, the water does not fully mix. Instead, it settles into layers, one resting heavily within another. In these places, dense brine collects in depressions, forming pools that ripple softly yet remain distinct from the surrounding sea. Their surfaces shimmer with a faint distortion, as though marking a boundary that should not exist underwater. These brines are created when salt deposits buried beneath sediments dissolve and seep upward, or when ancient evaporated seas left concentrated minerals trapped below. The resulting liquid can be several times saltier than typical seawater. Its density increases accordingly, causing it to sink and spread laterally along the seafloor like a slow-moving river confined within invisible banks. To most marine organisms, such environments are lethal. The high salinity disrupts cellular balance, drawing water out of tissues and overwhelming metabolic processes. Oxygen levels within these pools are often extremely low, sometimes absent entirely. Methane and hydrogen sulfide may accumulate, further altering chemistry. Fish that stray too far into the brine can become immobilized, their movements slowed until they drift motionless at the boundary. Yet life gathers at the margins. Where brine meets ordinary seawater, sharp chemical gradients create opportunities. Microbial communities thrive in these transition zones, drawing energy from compounds released by the dense fluid below. Bacteria form thin mats along the edges, their metabolic pathways adapted to conditions that would challenge most surface organisms. Small crustaceans and specialized worms inhabit nearby sediments, feeding on microbial growth sustained by the seep. In some regions, these pools appear almost like submerged lakes, with defined shorelines and gentle internal currents. Instruments lowered into them encounter resistance, as though passing through a liquid within a liquid. Sediments at the bottom of brine basins can preserve organic material with unusual efficiency, slowed in decomposition by the absence of oxygen. Such conditions offer glimpses into ancient biological records, quietly archived beneath layers of salt. The distribution of these systems remains uncertain. A handful have been documented in areas such as the Gulf of Mexico and the Mediterranean, yet vast portions of the deep sea floor remain unsurveyed in detail. Their formation depends on specific geological histories, including evaporite deposits and tectonic pathways that allow fluids to migrate upward. Whether similar lakes exist undiscovered in other basins is still an open matter of exploration. Stability over time also varies. Some brine pools appear persistent across repeated observations, maintaining distinct boundaries for years. Others may shift gradually as surrounding sediments move or as chemical gradients evolve. Within the broader motion of the ocean, these dense rivers and lakes occupy a delicate equilibrium, suspended between isolation and integration, shaped by processes unfolding far beneath the visible floor of the sea. Beneath layers of sediment that appear still and undisturbed, vast reservoirs of methane rest in quiet suspension. The gas is often locked within crystalline structures known as hydrates, where water molecules form cage like lattices around methane under high pressure and low temperature. In this state, it remains stable, embedded in the seafloor like frozen breath held for centuries. Conditions at depth maintain this balance, yet it is not permanent. A subtle rise in temperature or a shift in pressure can weaken the structure. When stability falters, methane escapes. Sometimes it seeps gradually, forming streams of tiny bubbles that ascend through the water column in delicate chains. At other times, destabilization occurs more abruptly. Sediments, fracture, and larger volumes of gas surge upward in concentrated plumes before dispersing. These releases do not unfold in isolation. Microbial communities inhabit the sediments where methane accumulates, forming intricate networks of consumption. Certain archaea oxidize methane anaerobically, partnering with sulfate-reducing bacteria in tightly coupled metabolic exchanges. Together, they convert much of the gas into dissolved bicarbonate and sulfide before it reaches open water. This biological filter reduces the amount that escapes into the ocean above. Even when methane enters the water column, additional microbes continue the process. Aerobic bacteria oxidize dissolved methane, transforming it into carbon dioxide. In many cases, only a fraction of the original release approaches the surface. The depth at which emissions occur plays a significant role in determining how much ultimately enters the atmosphere. Yet, uncertainty remains. Vast areas of continental margins contain hydrate deposits, whose total volume is still being estimated. Warming ocean temperatures could alter stability zones over long timescales, potentially increasing release rates. Submarine landslides, triggered by sediment weakening, may liberate stored methane in sudden pulses. Geological records suggest that large scale methane releases have occurred in Earth's past, associated with periods of climatic transition. Monitoring these regions requires a combination of seafloor observatories, seismic surveys, and chemical sensors capable of detecting subtle changes in gas concentration. Autonomous vehicles map bubble streams rising from the sediment, while cores extracted from the seabed reveal hydrate layers hidden below. Time series data accumulate slowly, offering glimpses into patterns that unfold over years or decades. Methane beneath the seafloor represents both an energy reservoir and a climatic variable. It exists in a delicate equilibrium shaped by pressure, temperature, microbial activity, and geological movement. Most of the time, the system remains stable, the gas confined within its crystalline lattice. When shifts occur, they are absorbed in part by biological processes and in part by the vastness of the ocean itself. The full balance between storage and release continues to be traced carefully, one measurement at a time. In the stillness of certain deep slopes and ridges, delicate frameworks rise from the seafloor, pale and intricate. They resemble forests in miniature, yet they grow without sunlight and without the vibrant colors associated with shallow tropical reefs. These are deep sea corals, some of which began forming long before recorded human history. Unlike their sunlit counterparts, these corals do not rely on photosynthetic symbionts. They feed by extending fine tentacles into passing currents, capturing organic particles and plankton drifting through the water. Each polyp secretes a skeleton of calcium carbonate or protein-based material, adding layer upon layer over years that stretch into centuries. Growth occurs at a measured pace, often only millimeters per year. Radiometric dating has revealed colonies thousands of years old. Certain black corals and bamboo corals preserve growth rings within their skeletons, subtle bands that record chemical signatures of surrounding water. These layers act as archives, storing information about past ocean conditions, including temperature and nutrient shifts. In their slow accumulation, they hold a quiet chronology of environmental change. Because growth is so gradual, disturbance carries lasting consequences. A single contact from fishing gear can fracture branches that required centuries to develop. Recovery, if it occurs at all, unfolds over similarly extended timescales. Broken structures may provide temporary habitat for smaller organisms, yet the original architectural complexity takes generations to rebuild. How these reefs establish themselves remains an area of ongoing study. Larvae released into the water column must locate suitable hard substrate, often exposed rock along steep slopes or seamounts. Currents influence settlement patterns, concentrating larvae in some regions while leaving others sparse. Once anchored, colonies expand slowly, creating three-dimensional habitats that attract fish, crustaceans, and other invertebrates seeking shelter or feeding grounds. Over time, clusters of colonies form reef-like assemblages, though they differ from tropical reefs in composition and energy source. They do not rely on sunlight-driven productivity, but on organic matter descending from above or transported laterally by currents. This dependence links them indirectly to surface processes while maintaining a distinct ecological identity. The broader role of deep-sea coral reefs in ocean ecosystems continues to be clarified. They appear to function as biodiversity hotspots, supporting species richness disproportionate to their spatial extent. Some commercially important fish use these structures during parts of their life cycle. The reefs may also influence local hydrodynamics, altering flow patterns in ways that affect sediment distribution and nutrient exchange. Within the deep ocean's vastness, these corals stand as patient architects. Their presence reflects endurance rather than rapid expansion. Over centuries, they construct habitats branch by branch, preserving within their skeletons a record of waters that have long since moved on. Time in the deep ocean does not announce itself with sunrise or dusk. There is no gradual brightening, no fading glow to signal transition. The water remains constant in its darkness, and without the daily arc of light, the usual markers that guide biological rhythms lose their clarity. At the surface, circadian cycles are anchored by the predictable rise and fall of the sun. Hormones fluctuate in response to changing illumination, guiding periods of activity and rest. In the deep sea, that external cue is absent, yet organisms still possess internal clocks shaped by evolutionary inheritance. The question is how those clocks function when detached from the sky. Some species appear to operate in continuous motion, drifting, feeding, or searching without obvious pauses. Long-term observation through stationary cameras has revealed patterns that do not align neatly with 24-hour cycles. Activity may cluster around feeding opportunities rather than fixed intervals. In environments where food arrives irregularly, often in faint pulses from above, remaining alert can carry advantage. Other animals display subtler forms of rest. Certain fish reduce movement and settle lightly against the seafloor, their metabolic rates decreasing for extended periods. Crustaceans have been observed remaining still within crevices, antennae held motionless as if conserving energy. In these moments, neural activity may shift into states analogous to sleep, though defining that state precisely remains complex. Sleep, as understood in terrestrial mammals, involves characteristic brain wave patterns and reduced responsiveness to external stimuli. Measuring such patterns in deep-sea species presents technical challenges. Electrodes and monitoring equipment must function under pressure and within small bodies. As a result, much of what is known comes from indirect indicators, changes in posture, slowed reaction times, or cyclical gene expression linked to circadian regulation. Genetic studies suggest that many deep-sea organisms retain clock genes similar to those found in surface relatives. These genes continue to oscillate in rhythmic patterns even without light cues, indicating that internal timing mechanisms persist. Whether those rhythms correspond to true sleep cycles or simply regulate metabolic processes remains under investigation. In some cases, tidal forces may offer alternative temporal structure. Even at great depth, subtle pressure changes linked to lunar cycles can be detected. Certain species show periodic behaviors aligned with these fluctuations, suggesting that gravity and water movement provide cues where sunlight does not. Within this steady darkness, rest may take forms less distinct than those familiar on land. Instead of defined nights, there may be gradients of activity, moments of reduced engagement woven into continuous existence. The deep sea does not erase the need for restoration. It reshapes how that need is expressed, allowing life to maintain balance without reference to day or night. In laboratories on land, biological limits are often defined by what fails first. Cells rupture under pressure, tissues degrade with time. Organs, once damaged, rarely return to their original form. Yet, in the deep ocean, Certain animals seem to move along different rules, as though the boundaries drawn elsewhere have softened. Consider organisms with bodies composed largely of soft tissue, lacking rigid skeletons or thick protective shells. At depths where pressure would compress air filled cavities and distort protein structures, these animals persist without visible strain. Their cells contain membranes enriched with specialized lipids that maintain flexibility under compression. Proteins fold in configurations stabilized by subtle molecular adjustments, allowing enzymes to function where surface equivalents would falter. Instead of resisting pressure, their biology accommodates it, integrating force into normal operation. Regeneration presents another quiet departure from expectation. Some deep dwelling echinoderms and worms can reconstruct lost appendages, replacing complex structures through coordinated cellular growth. In certain species, even portions of internal organs can be restored. Stem like cells remain distributed throughout their tissues, ready to activate when injury occurs. Signaling pathways guide new growth, reestablishing symmetry and function over time. The process unfolds slowly, aligned with the measured pace of their environment. Longevity introduces further intrigue. A number of deep-sea animals exhibit negligible signs of senescence across extended lifespans. Growth may continue at reduced rates for decades, yet markers of cellular aging, such as telomere shortening or accumulation of oxidative damage, appear moderated. Some species of fish and invertebrates maintain stable reproductive capacity well into advanced age. Their metabolic rates are often low, reducing the production of reactive byproducts that can harm DNA and proteins. The mechanisms behind these traits remain under careful study. Genomic analyses reveal variations in genes associated with DNA repair, protein stability, and stress response. Enhanced chaperone proteins assist in maintaining correct folding under pressure and cold. Antioxidant systems appear particularly robust in certain long-lived species, buffering cellular components against gradual deterioration. Yet translating these molecular patterns into a comprehensive explanation requires sustained observation and comparative research. These biological departures do not suggest invulnerability. Deep sea organisms remain subject to environmental change and disturbance. Rather, they illustrate alternative strategies shaped by isolation, stability, and constraint. Over evolutionary timescales, natural selection has refined cellular processes to align with conditions far removed from surface norms. Within this concealed realm, life does not simply endure extremes, it reorganizes itself around them. The familiar rules of fragility, aging, and structural necessity are not discarded but reinterpreted. In the steady pressure of the deep, biology reveals capacities that expand the understanding of what living systems can sustain. In certain regions of the deep ocean, energy does not arrive in steady abundance, but in faint increments, rising slowly through layers of life. The foundation of these systems may rest upon chemical reactions within sediments or along fractures in the crust, where microbes transform inorganic compounds into organic matter. From this quiet beginning, nourishment passes upward with measured restraint. Unlike surface ecosystems fueled by constant sunlight, these communities depend on limited sources. Organic particles drift downward from distant waters, scattered and uneven. Chemical gradients supply energy in localized pockets. The result is a food web that unfolds at an unhurried pace, each transfer of energy marked by scarcity rather than surplus. Microbial mats form thin living films across rocks and sediments, converting available compounds into biomass. Small invertebrates graze upon these layers, extracting sustenance from bacteria that themselves rely on subtle chemical imbalances. Growth rates remain modest. Reproduction occurs infrequently. Life persists through efficiency, conserving energy at nearly every step. Higher in the chain, predators navigate an environment where prey encounters may be separated by long intervals. Some fish possess expansive sensory systems capable of detecting minute vibrations or chemical traces, enabling them to locate distant meals. Others rely on ambush, remaining still for extended periods before seizing an opportunity. A single successful capture can sustain an individual for weeks or longer. Because interactions are rare and dispersed, many feeding relationships remain inferred rather than directly witnessed. Stomach content analysis offers partial insight, revealing fragments of crustaceans, worms, or smaller fish. Isotopic signatures within tissues help trace the origin of consumed carbon, mapping pathways that are otherwise invisible. Yet the precise sequence of events, the moment of pursuit, or the exchange between predator and prey, often escapes observation. Energy efficiency shapes morphology and behavior. Some organisms exhibit reduced musculature, favoring slow movement over rapid bursts. Others develop expandable stomachs to accommodate infrequent but substantial meals. Metabolic rates align with the rhythm of availability, rising briefly during feeding and settling into extended conservation. In such systems, the removal of even a small component can alter balance significantly. With few redundant pathways, each species occupies a defined niche. The absence of one grazer or predator may ripple outward slowly, affecting energy distribution across years rather than days. The deep ocean's slowest food chains illustrate an economy of survival built on patience. Energy ascends in measured increments, filtered through organisms adapted to endurance. Though many exchanges remain unseen, their cumulative effect sustains life in regions where abundance is not assumed. Within this deliberate network, persistence replaces speed, and continuity emerges from restraint. Across vast stretches of the deep ocean, change arrives gently. Temperatures remain nearly constant. Darkness does not fluctuate. Currents move with steady persistence rather than seasonal turbulence. In such stability, the pace of evolution appears to follow a different rhythm. On the surface, shifting climates and variable resources exert continuous pressure on populations. Traits that improve survival spread quickly, while less favorable variations diminish. In the deep, environmental constancy can reduce the urgency of rapid adaptation. When conditions remain similar across centuries, selection may act more subtly, refining rather than reshaping. Many deep-sea organisms grow slowly and mature late. Some fish require years before reproducing, and certain invertebrates may live for decades before reaching full size. With extended lifespans come longer generational intervals. Genetic changes, even when advantageous, propagate gradually through populations whose turnover is measured in long spans rather than seasons. Low metabolic rates also influence mutation dynamics. Reduced cellular division can mean fewer opportunities for replication errors to occur. Combined with stable habitats, this may contribute to slower rates of genetic divergence in some lineages. Molecular clock analyses have suggested that certain deep-dwelling species accumulate changes at a measured pace compared to their shallow relatives. Isolation adds another dimension. Deep basins separated by ridges and currents can limit gene flow between populations. When exchange is rare, Small groups may evolve independently, guided by local conditions. In some cases, this isolation produces unique adaptations finely tuned to specific chemical or physical parameters. In others, populations remain remarkably similar over wide geographic areas, reflecting limited external pressure to diverge. Scientists continue to examine how resilience functions within such steady environments. While stability can buffer against frequent disturbance, it may also constrain flexibility. Species adapted to narrow ranges of temperature or chemistry could face difficulty if those parameters shift. Fossil records and genetic reconstructions indicate that some deep lineages have persisted with minimal morphological change for millions of years, suggesting enduring compatibility with their surroundings. Yet the deep ocean is not entirely static. Tectonic movement, episodic volcanic activity, and gradual shifts in global circulation introduce change over geological timescales. Evolution in this context may operate on broad arcs, responding to transformations that unfold across epochs rather than decades. Within these depths, natural selection continues its quiet work. It does not halt, but it proceeds with patience aligned to the environment it shapes. Traits are conserved when they remain effective. Variations spread slowly when they offer advantage. Over time, measured in centuries and millennia, life in the deep refines itself, guided less by rapid upheaval and more by enduring equilibrium. Far below the restless surface, water moves with a patience that escapes casual notice. There are no whitecaps here, no visible signs of motion, yet entire rivers of seawater glide through darkness along paths defined not by wind, but by density. These deep currents form a hidden circulation system, steady and immense. Temperature and salinity act as quiet architects. When surface waters cool near polar regions, they become denser and sink, beginning slow journeys across ocean basins. Variations in salt concentration further influence this descent. The combined effect, known as thermohaline circulation, sets water masses into motion along gradients that can persist for centuries. Once established, these flows continue largely independent of atmospheric influence. As dense water spreads along the seafloor, it carries with it dissolved oxygen, nutrients, and fine particles. In some regions, currents funnel through narrow passages between ridges, accelerating as they pass constricted terrain. In others, they drift broadly across abyssal plains, redistributing sediments grain by grain. Over time, this movement sculpts subtle features into the seabed, carving channels and forming sediment waves that reveal the direction of unseen flow. Ecosystems adapt to these patterns. Where currents concentrate organic matter, filter feeding organisms gather, extending delicate structures into the passing water. Sponges and corals orient themselves toward prevailing flow, maximizing contact with suspended nutrients. In quieter zones, finer sediments settle, creating softer habitats favored by burrowing species. The presence or absence of current shapes community composition as surely as light shapes life in shallower waters. These movements also influence chemical balances. Oxygen-rich deep water replenishes regions that would otherwise become depleted. Nutrients carried across basins contribute indirectly to productivity. When currents eventually rise in upwelling zones, far from their origin. Thus, deep circulation links distant ecosystems, weaving them into a planetary network operating beyond immediate perception. Mapping these pathways remains an ongoing endeavor, instruments anchored to the seafloor record velocity and direction at fixed points, while drifting floats measure temperature and salinity at depth. Yet vast areas remain sparsely sampled. The contours of underwater mountains, trenches, and plateaus redirect flow in ways that models continue to refine. Small variations in density can alter trajectories subtly, producing branches and eddies that escape coarse resolution. Within this concealed system, water that once touched polar ice may travel thousands of kilometers before rising again. The journey unfolds over decades or longer, steady and unhurried. Though invisible to casual view, these currents guide sediment, sustain life, and connect oceans into a single breathing body. Beneath the surface calm, motion persists, shaping the deep with quiet persistence. Age in the deep ocean is not always marked by visible change. There are creatures moving slowly across the seafloor today, whose beginnings trace back centuries. Their bodies carry time quietly, without the rapid cycles of growth and decline more familiar in shallower waters. Certain deep-sea fish have been estimated to live well beyond a century. Some invertebrates, including specific corals and sponges, persist for hundreds or even thousands of years. These lifespans are not simply extensions of surface patterns, but reflections of an environment where pace itself is altered. Growth proceeds gradually. Reproduction may occur intermittently. Energy is spent with restraint. A key factor appears to be metabolism. In cold, stable conditions, metabolic rates remain low. Cells consume oxygen more slowly, reducing the accumulation of reactive molecules that can damage DNA and proteins over time. With fewer metabolic byproducts circulating through tissues, the gradual wear associated with aging may unfold at a diminished rate. Life stretches not through haste, but through conservation. At the cellular level, repair mechanisms show intriguing characteristics. Some long-lived species exhibit enhanced activity of enzymes involved in DNA repair, correcting mutations before they accumulate. Proteins responsible for maintaining structural integrity remain functional across extended spans. Telomeres, the protective caps at the ends of chromosomes, do not shorten as rapidly in certain deep-dwelling organisms as they do in many surface animals. These molecular details suggest that longevity is supported by active maintenance rather than simple inactivity. Reproductive strategies also influence lifespan. Species that mature late and produce fewer offspring often invest heavily in individual survival. In environments where external threats are relatively limited and conditions change slowly, extending life can be advantageous. A long-lived individual may reproduce multiple times across decades, ensuring continuity without relying on rapid generational turnover. Despite these observations, explanations remain incomplete. Not all deep-sea species are exceptionally long-lived. Some follow life cycles comparable to those of shallow relatives. Variability across taxa indicates that longevity arises from a combination of environmental stability, genetic adaptation, and ecological role. Isolating the precise contribution of each factor requires careful comparison across diverse lineages. Studying such extended lifespans presents practical challenges. Direct observation over centuries is impossible, so researchers rely on growth rings, radiocarbon dating, and molecular markers to estimate age. These methods provide insight, yet each carries limitations. Uncertainties persist, inviting further investigation. Within the deep ocean, time gathers differently. It accumulates in slow increments within tissues adapted to endure. Longevity here is not dramatic, or accelerated. It is steady, aligned with an environment where change arrives gradually and survival favors patience. In that measured rhythm, life extends itself quietly, continuing far beyond the span commonly expected at the surface. At first glance, many deep sea creatures seem almost unreal, as though imagined rather than evolved. Eyes appear oversized or vanish entirely. Jaws extend beyond proportional balance. Bodies stretch into elongated ribbons or compress into compact forms without clear symmetry. Yet each contour reflects a response to conditions that differ profoundly from those at the surface. In the absence of sunlight, appearance loses much of its social and ecological weight. Bright coloration offers little advantage where color cannot be seen. Camouflage shifts from blending with reefs or sand to minimizing silhouette against faint ambient glow. Some species develop transparent tissues, reducing visibility from nearly every angle. Others adopt dark pigmentation that absorbs the limited light present, rendering them nearly invisible in dim surroundings. Vision, where retained, changes in emphasis. Large eyes gather scarce photons, maximizing sensitivity over resolution. Certain fish possess tubular eyes, oriented upward, tuned to detect the faintest trace of bioluminescent movement above. In contrast, many organisms abandon sight almost entirely, investing instead in alternative senses. Elongated antennae sweep through water, detecting chemical signals or subtle vibrations. Lateral line systems become finely tuned, registering shifts in pressure that indicate distant motion. Jaw structures often appear exaggerated, yet scarcity shapes these adaptations. When encounters with prey are unpredictable, the ability to seize and retain any available food becomes critical. Hinged skulls and expandable stomachs allow consumption of organisms nearly equal in size to the predator. Teeth curve inward, preventing escape in a realm where missed opportunities may not be quickly replaced. Body shapes reflect energy efficiency. Streamlined forms reduce the cost of movement through dense water. Gelatinous tissues decrease metabolic demand, requiring less energy to maintain. Some animals exhibit reduced musculature, drifting rather than swimming continuously. In each case, the external form mirrors internal economy. Darkness also relaxes certain constraints present in illuminated ecosystems. Without the need for visual signaling, elaborate patterns and vibrant displays diminish. Selection favors traits that enhance detection, capture, and conservation rather than aesthetic complexity. Over evolutionary time, these pressures sculpt organisms into silhouettes unfamiliar to human expectation, yet precisely suited to their environment. Comparative genetic studies reveal that many deep-sea species share ancestry with surface relatives whose appearances differ markedly. Small shifts in developmental pathways, influenced by habitat and resource availability, accumulate across generations. What seems alien often represents a series of incremental adjustments aligned with survival demands. In the deep ocean, form follows function with uncompromising clarity. Features that appear unusual are expressions of adaptation to darkness, pressure, and limited food. Familiar standards of beauty or symmetry do not guide this process. Instead, evolution refines structures according to necessity, producing bodies that reflect the quiet logic of life, sustained far from light. A vehicle descends carrying its own small sun. Without artificial light, cameras would record almost nothing, yet illumination changes the scene the moment it appears. Beams cut through the water and fall across organisms that evolved in near perpetual darkness. What unfolds next is rarely untouched behavior. Some animals freeze when exposed to bright light, holding position as if suspended. Others retreat beyond the edge of visibility, disappearing into water just outside the illuminated cone. Predators that rely on stealth may abandon a hunt once glare reveals their outline. Prey species sometimes react with sudden bursts of motion uncharacteristic of their usual pace. In each case, the act of seeing alters what is seen. Noise introduces a similar complication. Thrusters hum. Mechanical arms extend with faint vibration. Even the subtle resonance of onboard systems can travel through dense water farther than expected. Many deep-sea organisms detect movement through sensitive lateral line systems or mechanoreceptors tuned to minute shifts in pressure. The presence of a submersible may therefore register long before it becomes visible, prompting changes in direction or posture that escape interpretation. Time also constrains observation. Reaching depth consumes hours, leaving limited opportunity for study before ascent becomes necessary. Weather at the surface, battery life, and operational schedules compress viewing windows further. A behavior that occurs once each day or once each season may fall entirely outside the period of surveillance. Absence in the record does not necessarily indicate absence in life. Stationary landers, equipped with low-light cameras, have begun to extend observation over longer intervals. Yet even these devices introduce foreign elements into the environment. Their shapes alter local flow patterns. Their faint emissions, whether light or sound, may influence nearby organisms. Absolute neutrality remains difficult to achieve. As a result, much of what is understood about deep-sea behavior arises indirectly. Stomach content analysis hints at feeding strategies. Stable isotope ratios trace energy pathways. Scars and growth patterns suggest encounters between species. Genetic expression profiles reveal shifts associated with stress or reproduction. From these fragments, scientists reconstruct likely interactions, piecing together narratives that cannot be fully witnessed. The deep ocean thus resists complete transparency. Its inhabitants continue their routines largely beyond sustained view. Observers must accept that presence changes context, that clarity carries disturbance. With each carefully planned descent, researchers gather moments rather than wholes, glimpses rather than continuous accounts. Within this interplay of light and shadow, knowledge advances cautiously. The challenge is not simply reaching the seafloor, but observing without rewriting the scene. In the quiet that follows a vehicle's departure, behavior resumes beyond the lens, unfolding according to patterns that remain partly concealed. Far above the abyss, life at the surface produces a steady rain. Microscopic algae convert carbon dioxide into organic tissue through photosynthesis, forming the base of vast marine food webs. When these organisms die or are consumed, fragments descend through the water column in a slow drift, often described as marine snow. What appears delicate from a distance carries profound planetary significance. As particles sink, some are eaten and recycled in shallower layers, yet a fraction continues downward, slipping beyond zones of active decomposition. In the deep ocean, colder temperatures and slower biological processes reduce the speed at which organic material breaks apart. Carbon becomes incorporated into sediments or remains dissolved in deep water masses isolated from the atmosphere. This transfer, known as the biological pump, functions as a long-term storage system. Carbon that reaches the seafloor may remain buried for centuries or longer, especially where sediments accumulate steadily. Even dissolved carbon carried by deep currents can circulate for hundreds of years before returning to the surface. During that interval, it is effectively removed from rapid exchange with the air. The scale of this vault is immense. The deep ocean contains far more carbon than the atmosphere and terrestrial vegetation combined. Small changes in how efficiently organic matter sinks, or how quickly it is remineralized, can influence atmospheric concentrations over time. Thus, processes unfolding in darkness contribute quietly to climate regulation on a global scale. Chemical interactions within sediments further shape storage. Microbial communities consume organic material, releasing some carbon back into surrounding water while locking other portions into stable compounds. In certain environments, carbonate shells and skeletal fragments accumulate, forming layers that archive carbon in mineral form. Over geological timescales, these deposits may become part of rock formations, extending storage beyond biological cycles. Despite its importance, many aspects of this system remain difficult to quantify. The exact proportion of surface production that reaches abyssal depths varies by region and season. Warming temperatures may alter microbial activity, potentially accelerating decomposition before particles sink fully. Shifts in ocean circulation could influence how long deep water retains dissolved carbon before resurfacing. Researchers deploy sediment traps, chemical sensors, and deep moorings to monitor these pathways. Samples collected from the seafloor reveal changes in accumulation rates, while isotopic analysis traces the origin of stored carbon. Yet the vastness of the ocean limits coverage, and models must interpolate between sparse measurements. Within the quiet of the deep, carbon settles into a slower cycle. What begins in sunlight descends into shadow, entering reservoirs that moderate atmospheric change. The ocean does not announce this role, yet through steady transport and patient storage, it shapes conditions far beyond its surface, holding within its depths a significant portion of Earth's climatic balance. Each expedition returns with images and samples that expand the catalog of life by small but steady increments. Even now, after centuries of marine study, new species descriptions appear regularly in scientific journals. The deep ocean, vast and unevenly explored, continues to yield organisms that have never been formally recorded. Part of this pattern reflects simple geography. Large portions of the seafloor remain unsurveyed at fine resolution. Research vessels concentrate on specific regions, often guided by funding priorities or technological access. Between these targeted sites lie immense tracts where no camera has paused and no sediment core has been extracted. Within such spaces, entire communities persist without documentation. Isolation also contributes to discovery. Submarine ridges, trenches, and basins can function as barriers, separating populations over evolutionary timescales. In these enclosed pockets, species adapt to local chemistry, temperature, and pressure conditions. Genetic divergence accumulates gradually, producing lineages distinct from relatives elsewhere. When exploration finally reaches these habitats, the organisms encountered may represent forms that evolved quietly beyond earlier surveys. Technological advancement plays a role as well. Improved imaging systems reveal smaller and more delicate species that previous equipment could not resolve. Environmental DNA sampling allows scientists to detect genetic traces suspended in water, indicating the presence of organisms rarely seen directly. Each new method broadens the field of view, uncovering diversity that had been present all along. Some species, however, may disappear before formal recognition. Deep sea mining, trawling, and climate-driven changes alter habitats in ways not yet fully understood. Organisms with restricted ranges could decline or vanish without ever receiving a scientific name. In a realm where observation is intermittent, absence may go unnoticed. Taxonomic work itself requires patience. Collected specimens must be compared carefully against known descriptions, anatomical details examined under microscopes, genetic sequences analyzed for divergence. Distinguishing a new species from variation within an existing one demands rigorous evaluation. As a result, discovery unfolds methodically rather than dramatically. Estimates of total deep sea biodiversity vary widely. Some projections suggest that a significant proportion of species remain undescribed, particularly among invertebrates and microorganisms. The true number depends on how life is defined and measured, on the resolution of sampling, and on the pace at which exploration continues. Within the deep ocean, novelty is not a sudden eruption but a steady emergence. Each descent adds a fragment to an expanding record. What appears new to science has often existed for millennia, unobserved but not absent. As technology advances and surveys extend outward, the catalog grows, reflecting both the richness of the environment and the long interval during which much of it lay beyond view. Long before forests rose on land or animals moved through shallow seas, Earth was a planet of extremes. Oceans were darker, chemistry more volatile, sunlight filtered through an atmosphere unlike the one known today. In certain regions of the modern deep sea, echoes of those early conditions persist. Hydrothermal systems and chemically rich sediments provide environments where energy arises not from light, but from interactions between rock and water. On the early Earth, before photosynthesis reshaped the atmosphere, similar reactions may have supplied the first metabolic pathways. Minerals, such as iron and sulfur, could have acted as catalysts, encouraging simple molecules to combine into more complex forms. In the deep ocean today, comparable reactions continue quietly, offering living laboratories for processes that may once have been universal. Microbial communities inhabiting these environments rely on chemical gradients rather than sunlight. Their metabolic strategies, including the use of hydrogen, methane, and sulfide, mirror pathways thought to predate oxygen-rich conditions. By examining the genomes of these organisms, researchers trace evolutionary lineages that extend deep into time. Certain enzymes appear conserved across vast spans, suggesting ancient origins embedded within modern cells. Sediments at great depth also serve as archives. Layer upon layer accumulates gradually, preserving microfossils, chemical signatures, and isotopic ratios that record shifts in climate and ocean composition. Within these strata lie clues to past mass extinctions, volcanic episodes, and changes in atmospheric carbon. The deep seafloor, largely undisturbed by erosion or surface weathering, safeguards these records with unusual stability. Yet, translating present observations into narratives of origin requires caution. Conditions on the early Earth were not identical to those found today, even in the deepest basins. Temperature, ocean chemistry, and atmospheric composition differed in ways still being reconstructed. Laboratory simulations attempt to replicate primordial environments, testing how simple organic molecules might assemble under pressure and heat. Some experiments yield promising pathways, while others reveal unexpected complexities. Field studies continue to refine these models. Submersibles collect samples from vent systems and subsurface sediments, analyzing mineral surfaces that may resemble those of ancient crust. Geochemical measurements help estimate how energy flowed through early oceans. Each discovery narrows uncertainty while leaving room for revision. The deep sea does not provide a complete mirror of the past, yet it offers rare access to processes shielded from rapid surface change. Within its darkness, ancient styles of metabolism endure, and sediments accumulate with patient regularity. By studying these environments, scientists approach questions that reach back billions of years, seeking to understand how non living chemistry crossed the threshold into biology. Much remains unresolved, but the deep ocean preserves fragments of Earth's earliest chapters, waiting to be read with care. Every descent into the deep begins with careful calculation. Power reserves are measured, pressure tolerances are reviewed, weather forecasts are studied with attention that borders on ritual. Even before leaving port, limitations define the mission. Submersibles, whether crewed or remotely operated, carry finite endurance. Batteries drain steadily in cold water. Life support systems in human occupied vehicles impose strict time boundaries. A dive that takes hours to reach target depth must still allow for ascent, leaving only a narrow interval for exploration. Each minute spent observing or sampling is balanced against the energy required to return safely. Robotic systems extend reach beyond what direct human presence can tolerate, yet they introduce their own constraints. Manipulator arms, designed to collect samples or adjust instruments, operate through layers of hydraulic and electronic translation. Fine motor control that feels natural in air becomes deliberate and imprecise under pressure. Grasping a fragile organism without damage demands exceptional skill and often repeated attempts. Sediment clouds stirred by movement can obscure vision, complicating even simple tasks. Communication with deep vehicles travels through cables or acoustic signals, both subject to interference and delay. Operators interpret camera feeds that may flicker or distort. Small shifts in orientation can alter perception of scale and distance. The physical separation between control room and seafloor adds complexity to every maneuver. Repairs at depth are rarely feasible. If a component fails thousands of meters below the surface, retrieval becomes the only option. Replacement parts must be engineered to withstand pressure, corrosion, and temperature extremes. Each modification undergoes rigorous testing before redeployment. The cycle from malfunction to improvement can span years. Cost further shapes the pace of progress. Research vessels require crews, fuel, and specialized equipment. Launch windows depend on funding cycles and institutional priorities. Building new submersibles or upgrading existing platforms involves collaboration across engineering disciplines. Each advancement accompanied by careful certification. Incremental improvements accumulate slowly, reflecting both financial and technical realities. Autonomous vehicles offer promise, capable of operating without tethers across broader areas. Yet, autonomy brings its own challenges. Navigating complex terrain without direct oversight demands sophisticated algorithms and sensors. Battery life remains a constraint, and retrieval is not guaranteed if systems falter. Despite these boundaries, innovation continues. Materials science refines hull construction, imaging technology improves clarity, data storage expands capacity for recording extended missions. Still, the deep ocean exceeds current capability in many respects. Vast regions remain beyond routine reach, not because they are unknowable, but because access demands patience, resources, and precision. Technology has opened passages into darkness that were once inaccessible. Even so, the ocean retains distance. Each descent represents a negotiation between ambition and limitation, progress measured not in leaps, but in careful steps that gradually extend the frontier of what can be seen and understood. Much of what is known about the deep ocean does not come from continuous witnessing, but from traces left behind. A core drawn from sediment, a fragment of tissue preserved in chilled storage, a chemical signature recorded by a passing sensor. These pieces stand in for events that unfolded unseen. Direct sampling can alter the very systems under study. Collecting a coral branch may disturb organisms that rely on its structure. Extracting sediment disrupts microbial layers arranged over decades. Even careful suction devices stir fine particles that settle differently once displaced. In habitats where growth is slow and balance delicate, intervention carries weight beyond the moment of collection. Because of these sensitivities, long-term monitoring stations are deployed sparingly. Installing equipment at great depth requires complex logistics and substantial resources. Once in place, instruments must endure pressure, corrosion, and biofouling for months or years. Power limitations constrain how much data can be gathered continuously. As a result, sustained observation exists in select locations rather than across entire basins. In the absence of constant presence, scientists turn to models. Mathematical frameworks simulate circulation patterns, nutrient cycling, and population dynamics. These models incorporate measurements from scattered sites and extend them across broader regions. Satellite data inform surface conditions, while deep measurements calibrate subsurface processes. The integration of datasets creates a composite picture, refined as new information becomes available. Laboratory experiments offer another layer of inference. Organisms retrieved from depth are studied under controlled conditions that approximate pressure and temperature. Researchers observe metabolic rates, reproduction, and stress responses, then extrapolate findings to natural settings. Though such experiments cannot replicate every variable, they illuminate mechanisms otherwise hidden. Isotopic analysis provides insight into pathways of energy and matter. Ratios of carbon or nitrogen within tissues reveal dietary sources. Trace metals in sediment indicate past hydrothermal activity. Genetic sequencing uncovers relationships among populations separated by distance. Each method contributes a fragment, a point of reference within a larger mosaic. Certainty in this context emerges incrementally. A single measurement rarely resolves a question fully. Instead, patterns gain strength as independent lines of evidence converge. Discrepancies prompt revision, encouraging more precise instrumentation or alternative hypotheses. The process resembles assembling a landscape from scattered reflections, each glimpse adding contour and depth. In the deep ocean, direct observation remains limited by distance and constraint. Yet through careful synthesis of indirect evidence, understanding advances. Knowledge forms not from uninterrupted viewing, but from attentive interpretation of signs embedded in water, sediment, and tissue. Gradually, the fragments align, revealing structures and processes that, though rarely seen in their entirety, become increasingly clear through accumulation and patience. In research archives and laboratory notebooks, there are entries that remain open long after the expedition has ended. A sound without a confirmed source. A species observed once and never relocated. A chemical anomaly recorded at depth with no clear explanation. These notes do not signal failure, they mark continuity. Some mysteries endure across generations of scientists. Data collected in one decade may be revisited years later with new analytical tools, revealing patterns that were previously invisible. Yet, even with technological advancement, certain questions resist closure. The deep ocean operates on scales of distance and time that exceed individual careers. A hypothesis proposed early in a researcher's life may still be under examination at its end. Not all scientific inquiry resolves quickly. In environments where access is limited and observation intermittent, accumulation of evidence unfolds gradually. A single dive offers fragments. A second dive years later adds context. A third, perhaps conducted by another institution, refines interpretation. The process resembles sediment settling, layer by layer, clarity emerging slowly from persistence. Uncertainty, in this setting, becomes a steady companion rather than an obstacle. It encourages careful language, measured conclusions, and openness to revision. When findings remain provisional, they invite continued exploration rather than definitive statements. The absence of immediate answers fosters collaboration across disciplines, linking geologists, biologists, chemists, and physicists in shared inquiry. There is also a quieter dimension to unanswered science. Extended engagement with unresolved questions shapes perspective. It tempers expectation of rapid resolution. It fosters respect for systems that reveal themselves in increments. In the deep sea, where change may unfold over centuries, patience aligns more closely with reality than urgency. Young researchers entering the field inherit both discoveries and uncertainties. They build upon data sets that span decades, adding new measurements while acknowledging the limits of current understanding. In doing so, they participate in a continuum rather than a series of isolated breakthroughs. The deep ocean, with its vastness and restraint, reinforces this approach. It does not yield comprehensive narratives in a single expedition. Instead, it offers glimpses that accumulate over time. Each unanswered question becomes part of a larger framework, guiding future efforts without demanding immediate resolution. Within this ongoing dialogue between inquiry and environment, discovery is not defined solely by what is solved. It is also shaped by what remains open, by the steady willingness to return, to measure again, to reconsider. The deep sea teaches that knowledge expands through patience, and that the weight of mystery can coexist with progress, carried forward across years, with quiet persistence. At the edge of every completed study, new lines of inquiry begin to form. Data tables close, reports are published, yet beneath the conclusions, another layer of curiosity gathers. The deep ocean, measured in fragments, continues to invite questions that extend beyond the reach of current evidence. Estimates of total life at depth vary widely. Microbial communities within sediments, water columns, and subsurface rock may represent a significant portion of Earth's biomass. Genetic surveys reveal sequences that do not match known organisms, suggesting lineages still uncharacterized. Each sample hints at abundance, yet comprehensive counts remain elusive. Vast regions have never been sampled at the resolution required to capture their smallest inhabitants. Connectivity between distant ecosystems also remains an active area of investigation. Larvae drift with currents that traverse basins, potentially linking populations separated by thousands of kilometers. At the same time, geological features such as ridges and trenches may isolate communities long enough for divergence. Chemical plumes from hydrothermal systems disperse nutrients across surrounding waters, influencing life beyond their immediate vicinity. The balance between isolation and exchange shapes biodiversity in ways still being mapped. There are habitats detected indirectly, inferred from chemical anomalies or acoustic signatures that await direct confirmation. Subsurface aquifers beneath the seafloor may host microbial assemblages sustained by mineral reactions. Pockets of life could persist within fractured basalt or beneath thick sediment layers, largely inaccessible to routine exploration. Each new instrument, capable of probing deeper strata, broadens the field of possibility. Temporal scales add further complexity. Some ecological processes unfold slowly, with cycles extending beyond the duration of most research programs. Population fluctuations may occur over decades or centuries, making short-term observations incomplete. Climate change introduces additional variables, altering temperature, circulation, and chemistry in ways that may reshape deep habitats gradually. Certain aspects of the deep ocean may remain beyond definitive measurement. Technical constraints limit coverage. Economic considerations influence expedition frequency. Even with advancing technology, absolute certainty across such vastness may be unattainable. Yet this recognition does not diminish inquiry. Instead, it frames exploration as an ongoing dialogue with an environment that reveals itself incrementally. The questions driving investigation are not signs of deficiency, but markers of engagement. They guide funding priorities, inspire new engineering solutions, and encourage interdisciplinary collaboration. Each descent contributes partial answers while uncovering further layers of complexity. Within the quiet expanse below, life persists in forms and quantities still being tallied. Connections weave across distances not yet fully charted. Some features remain unseen, not concealed by intent, but by scale and depth. The pursuit continues, shaped by curiosity that adapts alongside expanding knowledge, accepting that understanding may grow without ever reaching complete closure. Beyond the range of our instruments and vessels, there are regions that remain physically and conceptually distant. The deep ocean does not yield completely to mapping or measurement. Even as charts grow more detailed and datasets expand, certain areas persist as broad outlines rather than precise renderings. Some environments resist mastery not through concealment, but through scale and condition. Pressure, remoteness, and cost define practical limits. A trench several kilometers below the surface cannot be inhabited or surveyed continuously. A hydrothermal field may shift subtly between visits, altering the context of prior observations. The ocean floor itself moves with tectonic forces measured in centimeters per year, gradually reshaping landscapes beyond immediate detection. Acknowledging these constraints does not diminish scientific effort. Instead, it clarifies the framework within which discovery unfolds. Knowledge develops within boundaries set by technology, funding, and physical law. Certain measurements remain approximate. Certain processes are inferred rather than directly witnessed. Precision increases over time, yet never reaches absolute completion. Science advances slowly in such terrain. Improvements in materials, sensors, and computation extend reach incrementally. A submersible descends a little deeper. A model incorporates additional variables. A genetic analysis resolves relationships once ambiguous. Progress accumulates through refinement rather than sudden transformation. Within this gradual movement lies a recognition that understanding is always partial. Each new finding rests upon earlier assumptions, themselves subject to revision. Data interpreted today may acquire new meaning under future frameworks. The deep sea, vast and dynamic, reminds researchers that conclusions are provisional, held with care rather than certainty. Mystery, in this context, does not signal inadequacy. It reflects the scale of the subject. The ocean covers the majority of the planet, extending across depths and distances that exceed routine access. That portions remain uncharted aligns with the magnitude of the environment rather than with a lack of inquiry. Acceptance of limits fosters responsible interpretation. It encourages clarity about what is known and what remains unresolved. It sustains curiosity without overstating claims. The willingness to articulate boundaries becomes part of the discipline itself, reinforcing trust in findings that are presented with measured confidence. Within the deep ocean, human reach encounters edges defined by pressure, distance, and time. These edges do not halt exploration, they frame it. Knowledge grows within their contours, expanding outward gradually. In recognizing that mastery is neither immediate nor total, science continues with patience, carrying forward questions alongside answers, aware that mystery can coexist with progress without diminishing its value. Long after the instruments are powered down and the research vessel turns toward shore, the deep ocean continues in quiet continuity. It does not announce conclusions. It does not clarify uncertainties. It remains as it was before observation began, vast and largely undisturbed by the brief presence of human inquiry. What has been gathered over decades forms a mosaic rather than a portrait, a chemical profile from one basin, a biological survey from another, a seafloor map traced in narrow swaths across immense plains. Each contribution adds definition, yet between these measured corridors stretch expanses where no direct record exists. The deep sea reveals itself in fragments, offering detail without totality. Most of it remains unseen, not because it withholds, but because access is constrained by depth, distance, and time. Lightless water extends far beyond routine exploration routes. Sediments accumulate in places no instrument has sampled. Currents move through passages not yet charted with precision. The absence of complete coverage reflects practical limitation rather than intentional concealment. In this sense, the ocean is not hidden, only unreachable in full. Its processes continue whether observed or not. Microbial communities cycle nutrients within buried layers. Geological forces shift tectonic plates beneath kilometers of water. Organisms migrate, reproduce, and expire in rhythms independent of documentation. The system functions without requiring acknowledgement. Scientific understanding advances through careful approach, yet it does not erase immensity. Even the most detailed bathymetric maps resolve features at scales that leave finer textures unrepresented. Even the most comprehensive biodiversity surveys capture a subset of living forms. Knowledge expands outward from points of contact, surrounded by margins that remain undefined. This incompleteness carries a particular clarity. It situates human effort within a broader context, acknowledging both achievement and scale. The largest environment on Earth extends beyond immediate grasp, not as a failure of investigation, but as a reflection of proportion. Oceans cover the majority of the planet's surface and contain the majority of its habitable volume. Comprehensive familiarity with such magnitude would require reach beyond present capacity. For now, the deep ocean remains mostly unknown, not in the sense of myth or obscurity, but in measurable terms of coverage and certainty. What is known rests upon careful measurement and repeated verification. What is not yet known occupies far greater area. And so, the ocean keeps its silence. It offers glimpses without surrendering entirety, continuity without summary. Within its depths, processes unfold at scales that exceed routine perception. The story assembled so far is substantial, yet partial. The rest endures beneath the surface, patient and vast, awaiting future encounters that will add new fragments to an ever growing, never complete understanding.